Viticulture and Enology and Food Sciences and Technology at the University of California at Davis. He's a founder of the Lab Genomics Consortium and is a founding member of the UC Davis Milk Bioactives and Functional Glycobiology programs. In 2010, Dr. Mills was awarded the Cargill Flavor Systems Award from the American Dairy Science Association. Sounds wonderful. And he's going to speak to us this morning. Well, you better give us, it's right there, right? Nutrition and the infant gut microbiota, health and disease in the first 1,000 days. Thank you.
Thank you, and thank the organizers for inviting me here. I like to say that I work on the two most important beverages to humans, and that is milk and wine. I didn't bring my wine microbial terroir talk today. Maybe that's for a different time. But I can tell you all sorts of things about which Californian wines have which microbes. We've been studying milk at UC Davis for about 10 years now on a large collaborative project at UC Davis. And the reason we study it is because when you ask yourself what food evolved to make a consumer healthy, there's really only one answer. Even though I try to convince my daughter broccoli evolved to make healthy. And actually, I've given this intro so many times over the past 10 years, my daughter's actually watched me give it. And when we actually had broccoli a while ago, she actually came to me and said, "But Dad, this didn't evolve to make me healthy." To which I responded with the normal parent, "Shut up and eat your broccoli." But human milk is the only food that evolved to make humans healthy. All other foods that we eat evolved for their own purposes, or we bred them for our own purposes. But of course, milk also evolved to shape the infant microbiota. So it evolved for two targets, in a sense. And we can't really forget that. Also, if you think about the evolution of lactation, it evolved under a serious amount of stress or pressure with a lot of constraints because, of course, every bit of energy a mom was giving away was at her own risk. And yet the infant had to get enough energy and protection to survive. And so this shaped over mammalian evolution to provide a multiplicity of functions in milk that we would argue folks are just beginning to touch the surface of. And there's many, many cases of molecules in milk that have double and triple functions that we're starting to understand. So it's a very complex fluid that is remarkably understudied, in our opinion. So what is it composed of? What's milk composed of when you look at it? Well, of course, it's mostly water, but there's a variety of macro and micronutrients. And of those, the largest chunk is lactose. That makes sense. Lactose is needed by the infant as an energy source. So the infant consumes lactose. Of course, the infant consumes lipid and protein as well. And so majority of what you find in milk is food for the infant, and that makes sense. A little more strange are these large oligosaccharides, human milk oligosaccharides, that I'll talk a lot about in the next coming slides, that actually the infant doesn't possess the metabolic or the enzymatic capacity to degrade. So when you think about that evolutionary process for a very long time over mammalian lactation, resulted in a constituent that's actually the third largest constituent in milk that the infant can't consume. So why would that happen? I'll get to that in a second. In terms of microbiota shaping elements, we certainly have identified and people have studied over the years a variety of elements in milk, immunoglobulins, lysozyme, lactoferrin, et cetera, even free fatty acids that are known to modulate microbiota or in a sense, mostly kill organisms or remove organisms. So we already knew that milk had this capacity in it. So let's talk a little bit more about the glycoconjugates in milk. So including in the lipid category and the protein category, and of course the human milk oligosaccharide category, are these large oligosaccharides that are attached to a lipid, attached to a protein, or free. And so mom is delivering a range, a constellation of glycan structures in milk that's quite complex. If we just look at the human milk oligosaccharides And this is a cartoon that just represents the kinds of linkages present. It's not that every milk oligosaccharide looks like this. There is a large amount of oligosaccharides in human milk, up to about 200 different species, which is actually remarkable when you think about the possible linkages that, the number of species that could emanate from the number of linkages that are present is far larger than 200. So nature selected the smaller amount of oligosaccharides that are present. If you look at them and you look in terms just of chain length, the predominance of the human milk oligosaccharides are smaller. DPs of 4 to 8 or so represent most of the oligosaccharides, but there are very large ones as well. And they've been studied at UC Davis by really a truly amazing glycochemist, Carlita Labria, who's created libraries after libraries of human milk oligosaccharides so that they can be identified via various mass spec and bioinformatic tools. And also, he's now shifted over to glycolipids and glycoproteins so we can profile all of those, and I'll show you some of that detail in a little bit. In humans, most of the oligosaccharides are fucosylated, so they have this fucose linkage, and a higher proportion is fucosylated than sialated, and this is different in different animal milk. So bovine milk, for instance, has more sialated oligosaccharides. Why make these? Why would 200 million years of mammalian evolution result in this large amount of glycan delivered in milk? Well, there's been a variety of studies and suggestions over the years, from neural development related to the sialic acid content, to immune development, to pathogen deflection, which is a relatively straightforward concept, because some of the human milk oligosaccharides look like some of the glycans on epithelial surfaces. So if you're flooding your GI tract with human milk oligosaccharides, then pathogens will think they're binding to an epithelial surface when they're actually binding mom's milk, and it gets flushed out. So that makes a certain amount of sense. But another option is that maybe they enrich specific microbes. Maybe they act as what those of us in the probiotic and prebiotic industries call prebiotics. Well, is there any evidence for that? Well, it turns out there's a variety of evidence for that. Large study by Penders et al. With about 1,000 kids used quantitative PCR to demonstrate that bifidobacteria are predominate in breastfed infants. And the key here is a breastfed infant. You can't have, once you introduce other kinds of foods, life gets a bit more confusing. And if you introduce formula even, life gets more confusing in terms of the data. Other studies using other techniques have identified that bifidobacteria with a large number of infants are predominant in the early stages, so the 1 to 3 months of breastfed infants. And there's actually some differences between regions of the world. This is a study that came out last year between Africa versus Northern European kids, and Malawian kids had a larger predominance of bifidobacteria in their feces than the Finnish kids. And of course, this is the study that everybody's been referring to. Yesterday, the work coming out of Jeff Gordon's lab definitely showed the difference in the microbial ecology of the developing infant or until 3 years old. And what he noticed is that, at least in that very early stage, a lot of the shotgun reads and the 16S reads mapped to bifidobacterium. So there clearly is data suggesting that this specific genera is showing up in infants at this time. Another study in Japan showed that not every kid ends up with a high amount of bifidobacteria. And they were doing a study where they were looking at moms and infants, and they noticed their infants kind of split into 2 categories, sort of a low BIF infant, this is measured after 1 month, and a high BIF contingent. And there's sort of a BIF no man's land here, except for this one spot. So why bifidobacteria? Why would nature have selected this organism to show up in this spot at this time? It seems to be part of a shared history of all humans. As a matter of fact, Jeff's data would suggest this is the one common diet we all have. We end up with the same microbiota, and then as we start to split into our different communities and different foods, we all end up with slightly different microbiota. Well, there are some clues, because bifidobacteria are also used as probiotics, so there's a fair amount of probiotics research going on out there. And there's some clues to suggest why they might be helpful at this stage of life. This is a Nature paper published by a Japanese group a couple of years ago, where they were actually just screening bifidobacteria to see which ones were more protective to, in a mouse model for E. coli infection. And they literally were just screening different strains of bifidobacteria that they had, and they noticed a particular B. longum strain was quite protective of an 0157H7 challenge, whereas other strains were less protective. So they went and they sequenced and they looked at, well, what are the differences between the strains that are protective and the strains that are not protective? And it turns out the strains that were protective simply had some sugar catabolism genes, in this case so it could consume fructose, and that the presence of those genes also correlated with the presence of the common end product of bifidobacteria, which is acetic acid. And so there was an increased amount of acetic acid, and that itself, they showed quite nicely, is protective in terms of barrier function. So we have, in a sense, a reasonably simple rationale, if we can follow a simple one, that if we can just get bifidobacteria to grow on a specific sugar in situ, that the production of acetic acid will be protective. But of course, that's what could possibly be happening inside babies with mom's milk, because mom's delivering these very unusual sugars. So we've been interested at UC Davis in trying to understand the mechanism of milk shaping the infant microbiota. And so one of the questions we've been asking is, do the different mother milk types influence the microbiota that exists, and maybe influence the predominance of bifidobacteria? To get into that, you have to understand different types of glycans that genetically moms can deliver. So mothers can be a secretor, which means they have a FUP2 gene, wild-type FUP2 gene, which essentially allows them to deliver a 1,2 fucose linkage on a variety of glycoforms. And if they're a non-secretor, they can't deliver that. And you can, of course, categorize moms genetically by this secretor, non-secretor, just like you can do blood types. About 20% of the United States population is non-secretor, so it's not a higher percentage. Different parts of the world, there's different percentage non-secretors. And so we've been interested in understanding if that protects. Now, this was led by some data generated quite a few years ago by Ardith Morrow and David Newberg, who followed children, in this case it was in Mexico, and they basically typed the mom's milk and then just measured the incidence of diarrhea in the kids, and noticed that basically the children who were getting secretor mom's milk had less incidences of various, either Campylobacter, calicivirus, and just general diarrhea, suggesting that being a secretor mom and delivering that 1,2 fucosyl linkage on your glycoforms is more protective. We had a discussion about this at dinner last night, and just because things are grouped together doesn't mean that we can necessarily associate them, although I think the mechanism for this one is a little easier to understand. So we need mechanism. We have to go and find mechanism for why this might occur. And so my lab has been involved in looking at that a lot and trying to find out, well, first, who actually grows on human milk oligosaccharides? If we actually remove everything and test it, who can grow? This is an experiment that was carried out by Angela Markoval and Justin Sonnenberg's lab, where they actually put one of these sugars, lactolan neotetrose, in the drinking water of some notobotic mice that were co-colonized with Bacteroides theta or B. infantis. And as soon as they put LNNT into the drinking water, all of a sudden, B. infantis was predominated inside that mouse. And as soon as they took it out of the drinking water, it went back down. When they put it back in the drinking water, it went back up. B. infantis is able to consume this sugar in a competitive way, even though, as Angela and Justin had shown in this paper, Bacteroides has wonderful systems for degrading human milk oligosaccharides and growing on it. So it's very complex systems that are available to Bacteroides to consume these complex substrates. Even so, B. infantis can outcompete it in situ. So we were interested in understanding, well, which oligosaccharides does B. infantis or the other bifidos actually consume? So if you throw human milk oligosaccharides at them, what do they consume? This is some work done by Ricky Lacasio in my lab, also in Carlito's lab, quite a few years ago. This represents the proportional amounts of different mass to charge ratios, and you can think of these essentially as DP, degrees of polymerization, counting on up of pooled mom's milk, not individual mom's milk, but pooled mom's milk. And you can see most of it is represented by a bolus of oligosaccharides with lower DPs. And when we grew B. infantis on it, B. infantis seemed to target those lower DPs and just vacuum them right up. Whereas some of the organisms that didn't grow as well, B. breve and B. longum, just nibbled away at what was LNT, the first degree of polymerization, suggesting some rationale for who might show up in the intestine in the same time. Now, Carlito's lab, of course, can figure out isoforms, and so they can take any one of these mass to charge ratios and split it up by nano-LC and annotate the different isoforms. So we're now profiling all the different bifidobacteria that we have by these kind of heat maps, where we show a specific isoform on how well it was consumed or not consumed. And so we're mapping whole reams of bifidobacteria and seeing if this maps back to the kinds of in vivo data we've seen out of the feces, so we can start to understand it. And with this kind of tools in place, both looking at it from in a culture versus the feces, you can actually start to really describe the mechanism by which a particular feces might be dominated by a particular bifidobacteria. All right, what about the genome functions necessary for this? Of course, we have different growth phenotypes, so we would expect different genome functions. We've sequenced, a couple of years ago, we sequenced a couple of different forms. This is an adult longum, and this is our type strain, B. infantis. We've now sequenced about 40 of both. If you're looking for genes related to the degradation of human milk oligosaccharides, you're going to be looking for fucosidases, sialidases, and a variety of other ACEs that are necessary to pick apart these complex structures, as well as transport mechanisms, either for the monomers or for the large oligosaccharides. And at least in B. infantis, anyway, it was wonderfully represented by pretty much a single cluster that had everything you could imagine you need to consume a human milk oligosaccharide, from the fucosidase, from the glycosyl hydrolases to binding proteins that are necessary to bind the oligos to help transport them into the cell. And we've done studies on all aspects of this. We've done proteomics to understand which genes, at least on the cell surface, or which proteins are expressed on the cell surface when you grow a cell on human milk oligosaccharides versus when you grow it on, say, other prebiotics like galacto-oligosaccharides or inulin. And different mechanisms are deployed for these different sugars. And human milk oligosaccharides, in a sense, turn on different sets of genes that might be diagnostic of a child, if it's actually predominating inside a child, but they might be diagnostic of a BIF that is growing inside a child. So these become biomarkers, in a sense, for us in our other clinical studies. This is a slide that sadly puts together two PhD projects into one slide. Two students, we've actually gone down and mapped through the glycosyl hydrolases necessary to pick apart human milk oligosaccharides in a variety of detailed work, so we know which fucosidases are necessary, et cetera, and which ones are expressed. And we end up with a model. And the model for B. infantis is over here, where you end up with complex oligosaccharides are imported and picked apart by the various glycosyl hydrolases inside the cell. The model for B. bifidum, generated mostly in Japan, is more of an extracellular degradation that then brings in only certain components and leaves out other components. And so you can imagine from a competition and what you are feeding to your neighbor's perspective, this one, the sequestering version, might be more competitive in terms of leaving other sugars behind for other bifidobacteria to grow on. So we're trying to understand that now. But those aren't the only sugars available in milk. There's a whole bunch of glycolinkages. And we've been looking at particularly N-linked glycoproteins. And we identified a variety of bifidobacteria are able to actually cleave the glycans off of N-linked glycoproteins, of which a variety are present in human milk. And you can just see it by a gel shift assay if you're incubating a proxy protein, RNase B, with various bifidobacteria. And we cloned and identified the genes involved And it turns out there's a specific endoglycanase that can cleave at the chitobiose core, sort of the base of the tree, and release the glycans. It's actually heat stable, too, so it turns out to be a food processing enzyme at this point. And so we can end up with an organism that can not only access the free large glycans, but also cleave off the glycans from the human milk proteins that are there. So you can imagine how this would give it an added ability to colonize that niche. So we end up with a situation where we have bifidobacteria competing with a variety of other organisms in the intestine for all these different glycans, and because it has this unique ability to grow on these glycans, it can compete well. Data I haven't showed you yet, I don't have time, is that actually when you grow them on human milk oligosaccharides, they actually bind better to epithelial cells. You can take the same cell, grow it on lactose, it doesn't bind as well as when you grow it on human milk oligosaccharides. And additional data that's being developed with Helen Raybould is that they actually produce soluble signals that tickle the enteroendocrine system. And that's some work that's being assembled at this point by Helen Raybould at UC Davis. So these complex milk glycans enhance bifidobacterial colonization. At least all the data that we've generated so far seems to support a rationale for why that would be. With the 2 minutes I have left, since this was a topic of discussion yesterday about what can actually be translated out of the knowledge that we have, I wanted to point out a couple of things. One is we're doing a study with Mark Underwood at UC Davis looking at premature infants. And the study is using a B. infantis strain that grows on human milk oligosaccharides and a B. lactis. It's actually B. animalis subspecies lactis, which is the most common probiotic out there in yogurts, which doesn't grow in HMO. It pretty much flatlines when you try to grow it on HMO. We wanted to understand, of course, can we somehow encourage bifidobacteria in these premature infants? We're not trying to see if we can prevent necrotizing enterocolitis because you need a big study to do that. We were just trying to find out, can we actually colonize these kids and get persistence of a probiotic at a longer state in these children who have very dysbiotic microbiota. Finally, we're translating all this by looking in bovine milk to identify common motifs. And just to leave you with one slide, about 3 hours south of Davis is Hilmar Cheese. Hilmar Cheese is the largest cheese plant in the world. They process 13 million gallons of milk a day. They make a wee bit of whey. And you can actually mine whey for some of these oligosaccharides. And you can imagine how much whey you could get if we could just prove that there's some helpful oligosaccharides there, and we just mine it the right way. We really could deliver something in near term. And so that's one of the things we're working on as well. So with that, I'd like to thank all the students and professors involved, the large group at UC Davis, and of course, all the funding agencies. Thank you.
Thank you, Dr. Mills.
Okay.
Any quick questions? Yes. Go ahead, Marty.
That was really a terrific talk. Can you, the ratio of the fucosylated to the silated HMOs is quite interesting. One of the questions is, how dynamic is it comparing early nursing to later nursing, and the variation across mothers? Is it constant per mother? And finally, is there any feedback from the baby to the mother?
The latter question I don't know. That's a great question, and I don't know the latter question in terms of feedback. In terms of the oligo content delivered, we think mothers are going to group by secretor, non-secretor, because I think secretor moms are going to be in the early results that Carlito has generated. The early moms, or the secretor moms, are delivering more glycan, as far as we can tell. In terms of persistence over lactation, there's been a lot of debate over this, and it depends on how you measure it, because of course the child is consuming more oligosaccharides more milk over lactation. It's a very small child at first and a larger child later. And so actually the amount of oligosaccharides per volume of milk goes down, but the amount of milk that the child's consuming goes up. And so it's a little confusing in terms of how much oligosaccharide the child is actually delivering. I don't know the data on the amount of fucosylated variation, at least over lactation. I think it remains somewhat stable, as does the neutral oligos in general, which includes the fucosylated. stay somewhat stable over lactation. There's a study that Bruce German and Carly Labrie are doing with Andrew Prentice, working on the Gambia, and they showed that actually over seasonal cycles, where these moms are either go through a dry part of the year where they actually eat more, or a wet part of the year where they actually eat less, their milk changes, but the glycans stay the same. And so that this is, in a sense, a protected delivery device from a mom's perspective. So, but that's as much as I know at this point.
And finally, the polymorphism between secretors and non-secretors is well conserved. So presumably there's some value in being a non-secretor mom. What are your thinking there?
So I get this question a lot. There's a variety of pluses and minuses to being a secretor or a non-secretor. If you're a non-secretor, you also might be less susceptible to various diseases. And I mean if you genetically are a non-secretor. So the characterization was that those were the warriors because their wounds would heal faster if you were a non-secretor. How much it benefits you as a baby if your mom is a non-secretor, I'm not sure.
It may be an alternative benefit.
Yeah, there's something we haven't figured out.
Yeah, let's save questions and we'll have some time later in the session.